The first step of DNA replication is initiation. Initiation begins from a specific point called the origin. In order to initially open the strands, an initiator protein, DNA-A, binds to one flank of the origin called the DNA-A box. This causes the other flank of the origin to open, separating the two strands. After the double strand is opened, DNA-B helicase and DNA-C bind to either side of the opening or replication forks. Next, DNA-G primases bind to the same sites. DNA-C is then released, creating the primosome complex of a helicase and a primase, and starting the next phase of DNA replication. Elongation. Elongation begins from the single origin point. Elongation then proceeds bidirectionally to create two new circular chromosomes. Numerous proteins are involved during elongation. DNA-B helicase unzips and separates the double-stranded DNA. This creates both a lagging and a leading strand. Single-stranded binding proteins prevent the single DNA strands from reannealing. DNA-G primase synthesizes a single RNA primer for the leading strand. DNA polymerase III continuously adds nucleotide bases on the leading strand throughout elongation. The complementary lagging strand is synthesized in short segments called Okazaki fragments. This is because elongation can only take place in the 5' to 3' direction. As the strand is unwound and the lagging strand is revealed, primase must add a new primer for each Okazaki fragment. DNA polymerase III synthesizes each short fragment after the primer is placed. DNA polymerase I removes each RNA primer from the newly synthesized strands. DNA ligase connects the Okazaki fragments to create the whole complementary strand. The final stage of DNA replication is termination. As elongation is completing, the replication forks reach the termination sequences. TUS proteins attach to the termination sequences to slow the replication forks to a full stop as they meet 180 degrees from the origin. At the end of replication, both circular daughter chromosomes are joined at a single point opposite the origin. The protein, topoisomerase IV, separates the attached daughter chromosomes. This creates two whole semi-conservative circular chromosomes. and ends DNA replication.